nice. We've looked at the structures in lab, so I can breeze through some of the slides because a lot of it is just anatomy based. Okay, but the way I like to see the digestive system, I think, like I told you in lab, is like, I kind of imagine it as like a big tube, okay? So what goes in, you know, must come out, but they're two different substances, and it's your digestive system that picks and chooses what to absorb, okay? So what's involved in digestion? Okay, so first you have to ingest things, right? You have to take it in. So that's food and liquid, okay? And then you have to pull it or push it through the tube, which is called propulsion, okay? And peristalsis is like waves of smooth muscle contraction to allow the product to move through the tube, okay? Okay, then we have two different kinds of digestion that we need to be aware of, okay? We have mechanical digestion. What does that kind of sound like?
Chewing.
Chewing, right? Yeah. So it's breaking things down into smaller units, more absorbable units that your body ultimately will be able to identify. Not just quite yet in the mouth, but later on down the tube, kind of a thing, okay? And then we have chemical digestion. What do we think is involved in chemical digestion?
Bile.
Say it again?
Chemicals, bile.
Uh-huh. Bile,
Saliva.
saliva, digestive enzymes, right? So things that we've already been talking about. So that's kind of the chemical component. All right, so we're mechanically breaking it down physically, and then we're utilizing enzymes to further convert it into substances that your body will identify as being either absorbable or not. And then we get absorption, okay? And that includes both nutrients and water, all right? So when we're talking about nutrients, we have three main classes of items that we ingest, right? What are the three main nutritional elements that we need in our diet?
Carbohydrates.
Okay, so we need carbohydrates first, or it doesn't matter what order. Carbs, what else? Protein and
Sugars.
fats. Okay, so those are the three elements that we're gonna be talking about in terms of what digestion of what starts where. Okay, so we're talking about carbohydrates, proteins, and fats. Okay, and then of course we need to eliminate waste, and eliminating waste is through defecation. Okay. And waste is typically leftover ions, water, and substances that your body chooses not to absorb. Your digestive system chooses not to absorb it, okay? There are things that we ingest like fiber that you just don't digest. It just goes straight through the tube, okay? Okay. All right, so when we're talking about the digestive system, similar to other body systems that we've talked about, we have main organs and then we have accessory organs. So I think of the accessory organs here as organs that are not part of the main tube, but they contribute, okay? So part of the main tube, the alimentary canal is the same thing as your GI tract, your gastrointestinal tract, your digestive tract, it's all the same, okay? Starting with the mouth, we've identified the pharynx, the three regions of the pharynx, the esophagus, stomach, small intestine, large intestine, we know is also called the colon, and then at the opposite end of the tube, we have the rectum and anal canal. So accessory digestive organs assist in digestion. So your teeth and your tongue are going to assist with mechanical digestion, breaking things down, okay? The gallbladder is going to secrete bile to assist with digestion. Your salivary glands are going to assist by secreting saliva. Okay. Your liver is going to secrete bile And then what does your pancreas secrete? Digestive enzymes, uh-huh. And we'll talk about one other substance it secretes as well as part of digestion. Okay. All right, so we need to address the different layers of the digestive system wall. Okay, we've looked at this in lab, okay, the different layers. So we're going from deepest to the most superficial, okay? Deepest, we have an epithelial lining, okay? Now, depending on where you are in the tube, the epithelial tissue will change, okay? And you guys need to be aware of when those changes occur. But in a nutshell, okay, on either end of the tube, either your mouth or your anus, which are both open to the outside environment, right? What type of epithelial lining do you think we'll find there? Stratified squamous epithelial tissue, right? Because you ingest things or you put things in your mouth that could be potentially sharp, especially, just things that you may take in. And then also, depending on bowel movements, there's just more activity going on at the ends of the tubes. So you need more protection, right? So you got more layers. So throughout the majority of the tube, okay, you're not going to have stratified squamous. We're probably going to switch it up to a simple form of epithelial tissue in order to promote absorption, which is what digestion is, okay? So the innermost layer then is the epithelial lining, okay? So you got epithelial lining. Superficial to that, we have lamina propria, okay, which we know gives something for epithelium to attach to. And then we have this layer of muscularis mucosa, okay, smooth muscle. Okay, then superficial to that, we identified the submucosa, which in this picture shows you these little yellow structures, which are representative for glands. And then we have a lot of blood vessels and lymphatic vessels and nerves in this submucosa layer. Okay. All right. Then we have the muscularis externa, right? Which we identified in lab as having two layers. It has an inner, what? Circular layer, okay? And an outer longitudinal layer, okay? So the circular layer, okay, just like in the stomach, circular, it's going to go around the tube and it's going to act to promote what we call segmentation, which is it's on another slide. But segmentation is breaking down product into smaller pieces, okay? So that's segmentation when you come across it. So the circular layer is going to allow the diameter of the tube to get smaller and bigger, okay, which can further break up product and allow for it to move along the tube. The longitudinal layer, because it's going all the way down the length of the tube, is most likely going to promote moving the substance through the tube, right? So like peristalsis or propulsion, okay? Because with structure comes function. We've known that since day one. So, and then most superficially, because we're talking about organs in the abdominal cavity, okay, we're talking about a serosa. Can anyone give me what the specific name of the serosa is in the abdominal cavity? Starts with a P. Peritoneum, right? So we're talking about the peritoneal serous membrane, okay? Adventitia would be for organs that are not within the abdominal cavity because they do not have a serous membrane superficially, okay? And I'll mention that briefly in a bit. Okay, so in between the circular and longitudinal layers, we have two nerve plexuses, okay? And this is unique to the digestive system, all right? So, just looking at the submucosal layer, okay, and noticing glands and blood vessels, okay, if that nerve plexus was stimulated, what do you think would happen?
Secretion.
Right, you would get glandular secretion, right? Most likely of digestive enzymes, mucus, whatever else, okay? The myenteric nerve plexus, okay, in between the circular and longitudinal layers is most likely gonna promote some sort of movement, right? Some sort of propulsion or segmentation, okay? Allowing for product to be further divided into smaller pieces. So the next slide, kind of just, there's a lot of text here, but all I want you guys to really get out of this is that your gut, okay, has its own nervous system, kind of like the heart does, okay? And it's called the enteric nervous system, all right? And the enteric nervous system is composed of those two nerve plexuses, okay? It makes up its own unique nervous system, okay? So, regardless of the nervous system, this enteric nervous system can function on its own, okay? Can it be influenced by the nervous system as a whole? Yes. And if we're talking about digestion, and we're talking about smooth muscle contractions, what part or what division of the autonomic nervous system do you think we're talking about? Parasympathetic, right? So the parasympathetic nervous system can control, can promote and or inhibit it, okay? But the enteric nervous system doesn't need the parasympathetic nervous system to function. It can still function independently on its own, okay? So if the myenteric nerve plexus is stimulated, we get increased contraction, increased muscular contraction, increased muscular tone, which is going to increase propulsion or peristalsis. It's going to move product through the tube. That's why that nerve plexus is located in between the two muscular layers, okay? Sphincter tone means it can help reduce the diameter of the tube or make it larger, okay? Promoting segmentation. Submucosal plexus, because it's in the submucosa, it's going to promote secretion from glands, which is going to promote absorption. Okay. All right, so that's just general concept from this. Okay, so let's circle back to the serous membrane then, the peritoneum, which we looked at, what, first week of class? We know the serous membranes have two layers, an outer parietal layer and an intervisceral layer, right? Because we're talking about the abdominal cavity here, we're talking about peritoneum. Visceral peritoneum is going to lay directly on top of the digestive organs like saran wrap, right? And then the parietal peritoneum is going to surround the abdominal cavity, right? In between the two layers is what?
Serous fluid.
Is fluid, which is housed in a cavity. Okay, nothing new here. And the fluid is designed to reduce what? Friction, right? So you can do your cartwheels and jumping jacks and whatever else, and your gut is okay with that. Okay, you're not going to damage anything. Okay. All right, so mesentery. The definition of mesentery is essentially a double layer of the peritoneal membrane, okay? And we saw some of this in the cats, okay? We weren't specifically looking for it, but we did come across it, okay? So what mesentery does is it holds organs in place that are in the abdominal cavity, okay? It can also store fat. So most mesentery looks fatty. It looks like a fatty blanket, okay? And then it provides a route for blood vessels and nerves to come in and penetrate the organs. Okay, so they're just giving you an example here of an organ and just showing you, for example, the liver and another organ. There's both posterior mesenteries and anterior mesenteries, okay, to help stabilize these organs in the abdominal cavity. They're not just sloshing around independently. They're secured, okay? Now, during development, some organs start off in the peritoneal cavity and they migrate behind it at the end, okay? So these are called retroperitoneal organs, okay? Examples of retroperitoneal organs include your kidneys, okay? Your pancreas is a good example. The beginning of your small intestine, which is the duodenum. So these all kind of go behind. And then your ascending and descending colon. Okay, so those are examples of retroperitoneal organs. Okay, so, again, just repetition here. Mesentery, double layer of peritoneum. There's dorsal and ventral, okay? Some of them we call ligaments, but again, ligaments is kind of a misnomer because it's not attaching bone to bone, which is what we're used to, okay? So let's give some examples. Ventral mesenteries, anteriorly, okay? The falciform ligament. Okay, we identified that in the liver. Anteriorly, the falciform ligament comes and stabilizes the liver against the anterior abdominal wall. Okay, so that's considered a ventral mesentery. Even though we call it a ligament, it's a misnomer, but that's what it is. Okay, the lesser omentum. The lesser omentum is a fatty blanket that attaches to the stomach. Can anyone have an idea where on the stomach? The lesser curvature, right? So the lesser omentum here is coming up and attaching to the lesser curvature of the stomach. Okay, stabilizing the stomach. Okay. And then dorsal mesentery, so posteriorly, we have the greater omentum. Now the greater omentum, essentially on the cat, was the big fatty blanket that just kind of covered all of the organs. So, let me just go back a picture here. So the greater omentum here, okay, it's considered dorsal because it attaches posteriorly in the cavity. So it's kind of odd because it's the most anterior mesentery that you're gonna see when you're looking at guts, but it's considered a dorsal mesentery just because it attaches posteriorly, okay? But greater omentum, cut on this picture here, it attaches to the greater curvature of the stomach and then comes all the way down over the organs and then curls underneath posteriorly to attach. Okay. So the jejunum and the ileum, we know the last two parts of the small intestine, they have mesentery proper. So when you spread out the intestines of the cat, you saw this kind of like clear membrane type material. That was mesentery. And in that was blood vessels and nerves. Okay. The transverse colon, okay, it's never really depicted on pictures, but it has its own mesentery called the transverse mesocolon. And then the sigmoid colon has its own mesocolon as well. Okay. So again, all just to provide stability for these organs in the large abdominal cavity. Okay, so now let's start at the mouth and then we'll go through all the structures and associated characteristics, okay? Again, a lot of this you guys already know the anatomy. Okay, so the mouth is considered the oral cavity. We already discussed it's got stratified squamous epithelium in there because, you know, there's sharp things and we need protection. It's open to the outside environment. Our lips and cheeks are formed by muscles that we've identified, orbicularis oris, okay, masseter, buccinator muscles we didn't really identify, but some of the musculature we did. All right. So as far as the mouth goes, the vestibule, okay, is the space in between your teeth and your cheeks, okay? So if you blow your cheeks up like a squirrel storing nuts for the winter, that's the vestibule, okay? Space in between your teeth and your cheeks, okay? All right, we've got a couple frenulums, and frenulums are just kind of tissue anchoring attachments. So you have a labial frenulum, one on the bottom and one on the top. It helps stabilize your upper and lower lip. You can palpate that with your own tongue. And then underneath your tongue, you have a lingual frenulum that prevents the tongue from going too far posterior. You don't want to swallow your own tongue, right? So the palates, we already know. We identified the hard palate. Posterior to that is a soft palate. And then it ends in a boxing bag type structure called the uvula. Nothing new here. The arches we didn't necessarily identify, but they're, again, boundaries based on the name. So you've got the palatoglossal arch, which is the first arch, okay? And that's going to give you a separation between the soft palate and the tongue. So it's like a boundary, okay? And then posterior to that, we have the palatopharyngeal arch, which signifies the end of the oral cavity and the beginning of the oropharynx. Okay, so two arches. All right, that's it for the mouth, okay? Tongue, we've looked at this. Filiform, fungiform, valley. Okay, we know filiform look like shark tails. They are what give your tongue the rough texture. Fungiform are a little bit more square shape like mushroom caps. Okay, and then vallates are the big ones posteriorly that make kind of like a V, inverted V shape. Okay, the sulcus terminalis, you don't have to memorize that, but it's a boundary that separates vallate papilla from your lingual tonsil. So when you stick out your tongue, you can't see your lingual tonsil, it's too far back. But you can see your vallate papilla. Did anyone try the close your eyes and put something on the tip of your tongue? Yeah? Who'd you do it on? Like just a friend or yourself?
My brother.
And how did it go?
He couldn't taste anything.
Couldn't taste anything,
Yeah.
right? Yeah. It's cool, guys. I'm telling you, try it. Yeah, it's pretty awesome. Okay. So teeth for mastication. Mastication, of course, chewing. Sounds like masseter, right? We're just talking about adult teeth. Okay, so we have 32 adult teeth, and if we break it into quadrants, which is what this is up here, times 4, okay? So top left, top right, bottom left, bottom right. You have 2 what are called incisors, okay? And the incisors are the flat 2 front teeth that you have, and they're used for like kind of grabbing and tearing, okay? They're flat, and they're used for that. 1 canine, okay, so you have 1 canine in each quadrant, that's the pointed tooth, and that's for tearing and ripping. You have 2 what are called premolars in each quadrant, so 2 small molars, and then you have 3 adult molars in each quadrant. Okay. Okay. So that gives you a total of 32 adult teeth. Okay, so as far as teeth are concerned, the basic anatomy of a tooth, the crown is what you see, okay? The neck is embedded in your gums, and the root sits in the only joint that you find in the mouth. What's the name of that joint? Gomphosis. Yep. Okay. So your teeth are made of a bone-like component called dentin, which is actually stronger than bone. Okay. And on the crown, it's covered with enamel, which is what makes it white. And then in the root, it's covered with what's called cementum. And then superficial to cementum in the root is the periodontal ligament, and that's what anchors your tooth in the socket. Okay. Gingiva is a formal name for your gums. So if your gums get inflamed, what condition is that? Gingivitis, right. And then dental tubules and pulp is basically the inside. Okay, you've got pulp inside where you have arteries and veins that can feed the tooth, and then you also have nerves. So anyone that's had a cavity, myself included, it's uncomfortable. You can feel it, okay, when you start to erode down into the pulp. And what's the cranial nerve that they inject at the dental office? If you have to have a cavity filled? The trigeminal. Mhm. Yep. So impress your dentist the next time you see it. Okay, the salivary glands. We've identified these. Okay, we've got the big one, parotid, which is right on top of the masseter on either side. And then we have submandibular and sublingual. Again, this is all parasympathetic. Okay, we're resting and digesting. Okay, so salivary glands are going to produce saliva. And saliva is mainly water, but there's also enzymes. Okay. There's some mucus, there's some antibiotic, antibacterial items that are in your saliva, which is good because, again, it coats the mouth, which is open to the outside environment. Okay, so moving on down the tube from the oro and the laryngopharynx, okay, we know that air and food can go into those passages. We identified the epiglottis, guardian of the airways, that's supposed to flap over the larynx when you swallow. Okay, so we're still talking about stratified squamous here, okay, for protection. Nothing new. And then we get down to the esophagus, okay? So the esophagus is a muscular tube that's important for deglutition. Deglutition is swallowing, okay? So when you identify the esophagus, it's a long tube, okay? But the inside of it is not just this big circular opening. It actually has all of these grooves in it, okay, with epithelial tissue that are lining the inside of this tube, okay? Okay, so we already know from lab that the esophagus joins with the stomach, and it's got to go through an opening in the diaphragm in order to get there. And what's that opening called? The esophageal hiatus, okay? Now, once it penetrates that esophageal hiatus, there is a sphincter that's at the base of the esophagus, right at the start of the stomach. So as substance goes into the stomach, that sphincter shuts so that it doesn't regurgitate or go backwards, okay? But, okay, there are some people that may have an esophageal hiatus that's a little loosey-goosey. It's not a nice perfect fit around the esophagus. And that can cause what's called a hiatal hernia, where you get digestive organs wanting to poke into that opening. Not good. Okay. You also can get people that have problems with this lower sphincter. Okay. So that sphincter may not work as well, especially those that have the hernia. And so you're going to get regurgitation of acidic stomach content back into the tube, and that's called GERD, gastroesophageal reflux disease. Okay, so these people, typically after they eat a meal, they have to sit upright for a good hour or more. So you can't just eat a big meal and then go take a nap. Not going to happen, because that's just going to promote reflux. And reflux is painful, and you don't want to erode the inner lining of your esophagus. Ouch. Okay, with all that stomach acid. Okay, so we're still stratified squamous for that reason too. Okay, interestingly enough, the muscularis external layer, so the circular and longitudinal layer of the esophagus, the first third of it, the top third of it, is skeletal muscle. So you have volitional control over what you're swallowing. You can choose to swallow it or you can spit it out, right? The bottom third is just smooth muscle. So once you get down to the bottom third, you don't have control anymore. But the middle is a hybrid. It's like half and half. Okay. All right. Okay. Hold on. Hold on, I just have something that I wanted to mention. let's go back to this slide really quick, because I breezed over it. Okay. As far as salivary glands go, okay, with saliva, okay, saliva has more than just water in it. It's got mucus, it's got digestive enzymes. It has a specific digestive enzyme that's called amylase, okay? That amylase, we typically call it salivary amylase because you find it in the mouth as a derivative of your saliva. So it's an enzyme, it's a digestive enzyme called amylase. And it starts to digest starches. What's another word for starches? Carbohydrates. So you're initiating carbohydrate digestion in the mouth. Okay, because amylase is going to start breaking it down. Okay. Grab the measure. Okay. Everybody good with that? Kind of a point that I forgot. Okay. All right. So, moving down into the stomach from the esophagus, when food, liquid, product, anything enters the stomach, it now changes to a substance called chyme. It's going to combine with stomach acid, mucus, and all goodies in the stomach, and it's going to become chyme. So now we're switching to simple columnar cells, okay? And we get a little bit of absorption here in the stomach. So we get alcohol, water, electrolytes, some drugs like aspirin. Alcohol is a good thing, so when you drink it, you don't have to wait like an hour for your entire digestive system to process it, because once it hits your stomach, then you can absorb it. Okay. We already know the regions of the stomach from lab. We know the characteristics, the curvatures. We know uniquely, the stomach has 3 muscular layers. So it has a circular layer and a longitudinal layer, but it has a third, deeper layer called the oblique layer, right? And that allows for churning in multiple different directions, okay? Get the product covered with stomach acid, mucus, all right? And the pylorus, we identified a few structures in lab. So the opening or the enlargement here is the antrum, okay? The canal is leading towards the opening, which is called the pylorus. And then the sphincter is the smooth muscle that's actually allowing chyme to exit the stomach and enter the first part of the small intestine, which we call the duodenum. Okay, so in the stomach, we'll talk all about this in a bit, okay? We've got chemical digestion being initiated, specifically of proteins, okay? So in the stomach, now we're starting protein digestion, okay? Liquefaction of food is basically just converting it into chyme. All right, so we'll talk about individual secretions here as we go along. Okay, so we identified this in lab. Gastric pits and gastric glands, okay? So chyme has to enter the gastric pits, okay? And gastric pits are lined, we'll talk about this on the next slide, with mucus cells, okay? So these gastric pits superficially have mucus cells. So when product enters, okay, mucus cells are going to continue to develop and produce and secrete mucus, again, to ensure that the environment within the stomach is manageable. It isn't overly acidic, okay? So once you go deeper, then you enter a gastric gland, okay? And gastric glands have special cells. And these cells are going to secrete things, okay? In this picture, okay, even deeper in the muscularis externa here, Because we're talking about the stomach, there's an oblique layer, okay? But remember the villus model that you guys are looking at in lab, does the villus model have an oblique layer? Because that's small intestine. You only have oblique layer in the stomach. Okay, so that's why you don't see it on that model. You just see the inner circular and outer longitudinal layer on that model, okay? Okay, so let's do a close-up of a gastric gland and talk about some of the different cells that we have in here, okay? So this is deeper to the mucus cells. The mucus cells are more superficial. So now we're getting deeper, and the first kind of cell we're gonna find is parietal cells. So parietal cells are gonna be these big blue guys, and they're going to secrete hydrochloric acid. They also secrete intrinsic factor, and intrinsic factor helps with your B12, vitamin B12 absorption, okay? And the one item in your diet that you eat that has a lot of B12, does anyone have an idea? A lot of people don't necessarily get it, depending on their choices. Meat, okay, steak, meat, big source of vitamin B12, okay? Okay, so hydrochloric acid is being secreted by the parietal cells. Then we have more numerous chief cells, which are purple. Chief cells are gonna release what's called pepsinogen, okay? And pepsinogen is an inactive form of a digestive enzyme. So pepsinogen only becomes active when it's in contact with hydrochloric acid. Then it turns into pepsin, okay? Because you don't want to have digestive enzymes starting to digest if there's nothing there. You only want them doing their job when it's necessary. So pepsinogen is neutral until it meets hydrochloric acid, and then it turns into pepsin in order to promote digestion, okay? And then finally you have these green cells at the bottom called enteroendocrine cells that we talked about before, okay? They're endocrine-like cells because they secrete a hormone. And the hormone is called gastrin. And we've talked about gastrin. So gastrin is secreted by the stomach, and what does it target? It targets, in general, the stomach, yeah. So it's secreted from the stomach, and it targets the stomach. And it promotes digestion, okay, we're just summarizing. And I'll mention that again here in a minute when we talk about a couple others. Okay, so how is it that we don't digest our own stomach, let alone eat other animals' stomachs, which some people do in their diet, like tripe, okay? So there's ways that protect us from digesting our own stomach and allow us to eat other stomach if we chose to, okay? So you have the mucus coat that really helps kind of resist a highly acidic environment. You've got tight junctions in the cells, okay, so that all that stomach juice can't seep into the lamina propria and start breaking down the inner lining of your stomach. You don't want that. And then you've also got epithelial cell replacement, so you've got stem cells that are hanging out in your stomach lining that replace themselves every week. Kind of crazy. And that's all to protect you. Okay. All right, so the small intestine. Small intestine is where we have the most digestion and absorption, the most, okay? And it's called small intestine more so for the diameter, not the length of it. The length of the small intestine is actually quite long, which I think you appreciated in the cat. You noticed the small intestine was quite long, and the large intestine was not as intense on the cat. So again, peristalsis is movement of product through the tube, okay? And then segmentation is interactive, like sphincter shutting of the tube in order to break the product into smaller pieces, segments, okay? And 3 divisions of the small intestine that you guys have identified in lab are the duodenum, the jejunum, and the ileum. Okay. So at this point, we're still digesting carbs, and we're still digesting proteins. Okay, so one thing we have not addressed yet is fat. Okay. All right, so this looks like the villus model in lab. Okay, so inside the small intestine, okay, you have circular folds, which are similar to the inside of the esophagus, where it's just these circular folds of tissue, and you've got epithelial tissue lining the inside. Okay, so food and liquid, chyme, if you will, because it's coming from the stomach, is going to come in contact with that epithelial tissue. Okay, so we're still talking about simple columnar here because we're talking about absorption, right? So we've switched to a single layer of cells. So those circular folds, okay, are going to force the chyme to kind of do these revolutions through it, okay, in order to promote more time for absorption. So on top of the circular folds, we have these finger-like projections that we identified, and we called them villi, or villus singular, okay? And then inside of the villi, okay, so outside of each villus is simple columnar epithelial tissue. We identified that on the lab. Then we have, inside each villus, okay, blood vessels, all right? So carbs and proteins are going to be absorbed directly into your bloodstream. That's why we got all kinds of blood vessels in those lacteals. That's where we're absorbing from, okay? It's going directly into your bloodstream, okay? But fats first go into, what are these green structures called? Lacteals. Okay, so fats first get absorbed by lacteals in the small intestine. Okay, so we're starting the digestion of fats in the small intestine, but the lymphatic system picks it up first and then processes it, makes it a little bit more user-friendly, and then hands it back to the small intestine. Okay. So, on top of the villi, which we can't really see in the model in lab, so on top of every single finger-like projection in the small intestine, we have even more villi, and it's called microvilli. And that's all to increase absorption. Okay, so think of like shag carpeting. If villi is shag carpeting, you've got shag carpeting on top of the shag carpeting. Oh my God, when I was growing up, my parents, we had the best green shag carpet. It was like grass. I loved that carpet. Anyways, love that carpet. Okay, so we got shag carpeting on top of shag carpeting, all to increase absorption. So with structure comes function, okay? And then we identified some glandular structures on the model, okay? One of them is Peyer's patches, and we identified those under the microscope as well as on a model, and that's part of MALT. So that's part of our mucosa-associated lymphoid tissue. Again, immune cells hanging out and waiting, just in case you've identified or absorbed something that you shouldn't have, and they can mount an immune response as needed, okay? So the enteroendocrine cells, we'll talk about shortly, are going to secrete hormones. Gastrin is one of them, okay? The duodenal glands, the Brunner's glands that we identified under the microscope, and they're representative on the model. They're not as pretty in the submucosa, okay? But those are going to secrete a bicarbonate mucus. So those Brunner's glands secrete a mucus, again, in order to temper the acidic environment, because the chyme that's coming from the stomach is highly acidic. So we need to calm that down so that we're not eroding the inside of our small intestine, where the most absorption is happening for digestion. Right. Okay, and then we just have general, what are called intestinal glands. We identify those as like little crypts here. And they're also kind of secreting digestive enzymes, antibacterials, kind of good flora and fauna, if you will, for your gut. Okay, like probiotic stuff. Okay. So let's talk about the enteroendocrine cells that I want you to know. Okay, so we already know gastrin. So what I want you to know is, again, the name of the hormone, where is it secreted, what's its target, and what happens when it gets there. Okay, just like the endocrine system. So gastrin is secreted from what organ? The stomach. And it targets what organ? The stomach. And it promotes digestion. It stimulates the plexuses. Okay, submucosal and myenteric plexuses. Stimulates them. Let's get moving. Let's secrete. Let's propulse. Okay. Okay. Another one we've already talked about, CCK, cholecystokinin. Okay. So cholecystokinin is secreted from the small intestine, specifically the duodenum. All three of these that I'm talking about here are going to be secreted by the duodenum, okay? Because that's where the most absorption happens. So it's going to be secreted by the duodenum, and it's got two targets. It stimulates the pancreas, and then it also stimulates the gallbladder. It's going to tell the gallbladder to secrete bile, right? Because gallbladder is a bile storage area. So it's going to tell the gallbladder, okay, secrete some bile. We're starting to digest and absorb, so we need some help. So bile comes in. And then for pancreas, okay, the pancreas is going to secrete what? Digestive enzymes. Those digestive enzymes are going to get back into the duodenum. How are those digestive enzymes going to get back into the duodenum? Through what structure in the pancreas that you identified?
The bottom.
That's a region on the organ. What specific structure inside of the pancreas did you guys identify?
Pancreatic duct.
It's the main pancreatic duct, right? So it's going to travel through there, and it's going to go boop, right into the duodenum, out the major duodenal, what?
Pellet.
Papilla. Yep. Okay. Okay, so secretin is another hormone secreted by the duodenum, but it only targets the pancreas, okay? And it's going to tell the pancreas, let's secrete some mucus. We need to mellow out the acidity, okay? So the pancreas is going to release bicarbonate mucus through the main pancreatic duct, out the major duodenal papilla, into the duodenum, okay, to make sure that the environment isn't too acidic. Okay, then the last hormone is called gastric inhibitory peptide, GIP. It's pretty much tells you what it does in the name, okay? So GIP is going to be secreted from the duodenum, and what do you think its target is?
Stomach.
The stomach. And what do you think it's going to tell the stomach to do?
Stop secreting gastrin.
Yeah, stop, slow down. So decrease, slow down your emptying into the duodenum. Yeah, so it's going to tell gastrin, take a nap, okay? Give us some time to absorb. Okay, so those are just the 4 hormones I want you guys to know for digestion. 2 of them we've already talked about. All right. Okay, so this is just driving home the fact that in the small intestine, okay, we have digestive enzymes that are going to digest carbs, proteins, and now fats. Okay, And lipase is one of the enzymes that breaks down fat in the small intestine. Okay. Okay, so the duodenum. Okay, we already know site of most absorption, receives digestive enzymes and bile. So it's receiving digestive enzymes from the pancreas, bile from the gallbladder and the liver. So Brunner's glands now we know are going to secrete a mucus to neutralize any kind of acid. Again, the major duodenal papilla we identified in lab, but we didn't identify this kind of enlargement here. And this is where the common bile duct and the main pancreatic duct come together. And it's called the hepatopancreatic ampulla. Ampulla enlargement, and it's telling you of what two organs are coming together. So it's the common bile duct from the liver and the main pancreatic duct from the pancreas. So that's a new structure. Okay, so the pancreas. You guys know head, body, tail. We know that it's both an exocrine and endocrine organ. Okay. Exocrine function is 90% of what it does, and that's the digestive enzyme part, right? So all of those acinar cells that we looked at under the microscope are what make and store those enzymes. Okay. Now, what I want you to remember is that the pancreas, when it secretes the enzymes, it secretes them as an inactive form first, kind of like pepsinogen. So it's going to secrete them as what we call zymogens. Okay? And they're only going to become activated when they get to the duodenum, because you don't want to digest your pancreas. Right? The pancreas is making digestive enzymes, but it's going to deliver it to the duodenum where digestion is occurring. Pancreas is an accessory organ. It's not part of the main tube, right? So zymogens are produced, they go through the main pancreatic duct, and they turn into active amylases, okay, just digestive enzymes in the duodenum. We also know the pancreas secretes mucus, okay, to neutralize the chyme because it's very acidic. So if secretin is secreted by the duodenum, then the pancreas is going to be triggered to secrete mucus. Okay? If CCK is secreted by the duodenum, then the pancreas is going to secrete digestive enzymes. Okay? And we already know the endocrine function for regulating blood sugar, the alpha cells and the beta cells, secreting insulin and glucagon respectively. This is not really, again, I'm just driving home here. You don't have to memorize all this. It's just saying that we're starting carbs. Carbs are green. We're starting carbs in the mouth, and we're continuing them in the small intestine. We're starting proteins in the stomach, and we're continuing them in the small intestine. Okay? We're starting fats in the small intestine. But first it's delivered to lacteals, to the lymphatic system for further processing before it's returned to the small intestine for digestion. Okay? Okay. So the pancreas is kind of a big deal for digestion. Okay? Because if you don't have pancreatic enzymes, 60% of your fat in your diet is not absorbed. And 30 to 40% of your protein and carbs are not absorbed. So those digestive enzymes are super important for successful digestion. Okay. So, and usually people that have a diagnosis of pancreatic cancer, it's not good in terms of your longevity. All right. Ulcers, just in general, okay, people used to think that ulcers are caused by stress. Okay, that's not true. Ulcers are a bacterial infection. Okay. and the bacteria starts to erode the inner lining of either your stomach or your duodenum, and that causes ulcers. That's what ulcers are. H. pylori, you don't have to memorize that, but that's a bacteria that you may have heard of that contributes to this eroding of your inner lining. So if somebody presents with symptoms of an ulcer, they can take an antibacterial, okay, over time, and it can heal. Okay, it's not caused by stress. High blood pressure, okay, yeah, that can be caused by stress. We've already learned that, right? Excessive amounts of cortisol, yes, can cause bad things to happen to the body. Okay, liver. Large filter, right? So it produces bile. It also helps us break down fats. The liver has so many functions, okay? I mean, it just so many things. It detoxifies us, number one. It processes our fats. It actually stores vitamins, yeah, and makes blood proteins. So it's kind of a big deal, all right? The porta hepatis, we've already looked at that in lab. Okay, it's 3 vessels coming together, the hepatic portal vein, okay, hepatic artery, which is feeding the liver as an organ, and then the common hepatic duct, okay, which is bringing bile from the liver to the duodenum. Okay, so that's the porta hepatis, which is just a system of 3 vessels. Okay, as far as gross anatomy goes, you guys have looked at this in lab, so we know lobes, falciform ligament, okay, ligamentum venosum, ligamentum teres, hepatic veins, again, drain blood from the liver itself as an organ. Across the front side, you guys identified the falciform ligament. Okay, that's part of that ventral mesentery that's going to stabilize it against the anterior abdominal wall. Okay, and then we looked at it microscopically too. Okay, so a lobule is the smallest microscopic anatomical structure in the liver, of which there are billions in the liver. Okay, so each lobule at each point has a triad of vessels. It's got a branch off of the hepatic portal. It's got a hepatic artery, which is going to feed it as an organ, and then it has a bile duct to deliver bile from the liver to the small intestine. Okay? So the branches of the hepatic portal vein, okay, are bringing in blood from digestive organs to the liver first for processing, before it goes back to the IBC and into general circulation to protect you against anything potentially pathological that you've ingested, right? So these portal veins connect to these channels that are called what? Sinusoids, right? Like, sounds like sinuses, which we know are channels that carry something. And all of these sinuses are going to collect at a middle structure that you guys identified in lab called what? The central vein. And inside of these sinuses are these little guys called what? Kupffer cells. And so they're just waiting to mount an immune response to something foreign that shouldn't be there. Okay. And so then all those central veins will get connected to a couple, usually a few large hepatic veins, and then that dumps into the IVC. Okay. Now, there's another PowerPoint that I have for you guys that's labeled hepatic portal system. I'm not lecturing on that, but if you need a little bit more information about the portal system, why it's there and what it does, that PowerPoint is there to assist you with a little it's just an extra resource. Okay, so the gallbladder. We know it stores bile, okay? This is where you can get gallstones if you get a little bit too much cholesterol. Can be quite painful. Okay. So coming straight out of the gallbladder itself is the cystic duct, right? And it joins up with the common hepatic duct to form what? To form the common bile duct, right? Mhm. So if you get gallstones, it can block the cystic duct, which can prevent the gallbladder from secreting bile, which can make digestion uncomfortable if you don't have enough assistance. And or you can develop gallstones, which can be very painful when you're eating. So people can have their gallbladder removed. You don't have to have a gallbladder. Does anyone know of someone that's had a gallbladder removed? Yeah, you don't have to have one. So they'll just go in, snip the cystic duct, take the gallbladder out, because the liver is also producing bile, okay? But it's just now you don't have a little storage vesicle that's giving you extra, right? So those people who've had their gallbladders out, they know how uncomfortable it is. So they're probably not gonna go to In-N-Out and have a 4x4 animal style with fries and a milkshake, because that's just gonna overload the system, right? It's gonna be really uncomfortable. So they learn what their tolerance is in terms of handling rich, fatty foods. And they just have to modify their diet a little bit, but they can still function perfectly fine. Okay, getting towards the end of the tube here, large intestine. So there's mainly absorption of water and electrolytes at this point. So most of your nutrients have already been absorbed through the small intestine, right? So large intestine now's main job is to start forming feces, form waste from the leftover products that were not absorbed, okay? So that's why you have these individual containers that you identified as what? Each one of these is a house drum, okay? So that helps move, again, product from house drum to house drum, because you've got to go against gravity up the ascending colon. Okay, so the cecum we know is the beginning of the large intestine. That's where the appendix kind of comes out here. They're showing you some mesentery in this picture, which is kind of nice. So you can see some sigmoid mesentery, little transverse mesentery. Okay, and the appendix, again, is just modified immune tissue, okay? Colon, you guys identified in lab, ascending, transverse, descending. You've got two flexures, which is the curves that they take. The tenia coli is smooth muscle. That's going to promote peristalsis. Okay. Epiploic appendages, we didn't identify that in lab, but they're basically just little fatty filled pouches here. That's all those are. Okay, and we know in the colon, under the microscope, there's a lot more goblet cells, right? So that's one of the characteristics. If you're looking under the microscope, this has got to be the colon, because there are goblet cells everywhere, versus the small intestine, not as many. Because the small intestine is getting mucus from the pancreas, right? So it doesn't need as many goblet cells. It's got assistance. Okay, and then we reach the end of the tube, okay? So we have an internal and an external anal sphincter, okay? Internal anal sphincter, smooth muscle. External anal sphincter, skeletal muscle. Well, that's convenient, okay? So if you feel like, or your body tells you that you need to void, okay, or have a bowel movement, then you're gonna feel that smooth muscle start to relax. But fortunately, you have an external anal sphincter, so you can pucker up until it's appropriate to get to the facilities, right? It's amazing to think of, like, who designed this? When you stop and think about it, it's like, wow. With structure comes function, yeah, so. And this little guy here, we're not identifying it, you don't have to memorize it, but super duper helpful, okay? Because he allows you essentially to fart and you don't poop your pants. So, another fun fact. Yeah. Okay, is that it?